Welcome to this lecture series, Crop Anatomy Lecture Series 9. In this lecture series, we'll be looking at the tissue systems. What are the tissue systems? All the tissues of our plant which perform the general function, regardless of position or continuity in the body, may be considered to form together a tissue system. So for tissues coming together, carrying out similar functions, are termed tissue systems. A vascular plant comes into existence morphologically from a single unicellular zygote. By further development, the zygote develops into the embryo and ultimately into the mature sporophyte or plant body. In higher plants, division of labor exists, and due to this, the tissues are arranged in system called tissue system. Each system usually consists of only one tissue or an association of tissues which perform common function and have the same origin, which means that some tissue systems are made up of just one tissue. Some tissue systems are a combination of tissues performing a common function or having same origin. There are three main tissue systems in the plant. We have the dermal or epidermal tissue system, we have the fundamental or ground tissue system, and we have the vascular tissue system. An example of a dermal tissue system comprises of the epidermis, which has the cuticular part. We have the upper epidermis, the palisade mesophyll, the spongy mesophyll, and the lower epidermis. Okay, we have in line with the stomata area the two guard cells that helps to close or open in order to aid the exchange of gases in and out of the leaf or the plant. The dermal system forms the outer protective covering of the plant and is represented in the primary plant body by the epidermis. However, during secondary degree growth, the epidermis may be replaced by another dermal system called the periderm. You remember the periderm in the last class about tissues. If you've forgotten, please go back and learn more about periderm. The epidermis usually consists of a single layer of cells which cover the whole outer surface of the plant in the entire body, except for certain small pores called stomata and lenticels. So the only area where the epidermis does not cover are the areas called stomata and lenticels. Epidermis is derived from two Greek words, epi, which means upon, and derma, which means skin, upon skin, skin upon. Okay? The epidermal cells may be somewhat irregular in outline, usually varying in shape and size, and arranged very close to each other, having no intercellular spaces among them. The cells possess a large vacuole and thin peripheral cytoplasm. The cells may contain leucoplasts, anthocyanins, and chromoplasts, but no chloroplasts except in guard cells. So generally the epidermis does not have chloroplasts except in areas where the guard cells are located. Okay, except inside the guard cells areas actually. So the wall's epidermal cells have an unevenly thickened wall. This additional thickness is due to the impregnation of suberin or cutin. I showed you the cutin, right? The suberization or cutinization of the wall protects the epidermis from mechanical injuries and prevents loss of water from the plant. In case of roots, the outermost layer is called epiblema, piliferous layer, or rhizodermis. So please note these differences in terms of the covering of the plant, of the shoots and the roots. What are the functions of epidermis? It is primarily a protective layer which helps in the protection of the internal soft tissues against mechanical injuries. It prevents excessive evaporation of water from the internal tissues, serves in photosynthesis and secretion, acts as storehouse of water in many xerophytic plants. Some of the epidermal cells develop into secretory tissues of nectaries, the stomata of leaves and stems, and the absorbing hairs of roots. Okay? Now let's look at the stomata, which is a component of the epidermis, epidermal tissue. These are minute pores which occur in the epidermis of the plant. Each of the stomata remains surrounded by guard cells that are kidney or bean shaped. Stomata may occur on any part of a plant except the roots. Those epidermal cells bordering the guard cells are called accessory cells, those cells that are bordering the guard cells, the cells immediately after the guard cells. Okay, the guard cells are living and contain chloroplasts in them. They also contain a larger protoplasm than other epidermal cells. In the leaves of diocots, the stomata remain scattered, whereas in the leaves of monocots, they are arranged in parallel rows. Under normal conditions, the stomata remains closed in the absence of light and open in the presence of light. In other words, at night, the stomata remains closed, while at daytime, the stomata remains open. This could be due to the environmental changes, differences in weather temperature, which leads to increased transpiration activity at daytime and reduce or no transpiration activities or gaseous exchange at night. This is a diagram of the stomata. We have the epidermal cells, we have the accessory cells, okay, the subsidiary cells or accessory cells, we have the guard cells. These are the accessory cells, these are the guard cells. We have the chromoplast, we have where the chlorophyll is situated, which gives it that green coloration. We have the stomatal pores. This diagram is polylabeled. So these are the nucleus. Okay, these are nucleus. This is the stomatal pore, the opening for the stomata. These are the chloroplast, which gives it that green nature. Okay, all right. So what again, these are the guard cells. Okay, so we have the accessory cells, we have the epidermal cells. All of these make up the epidermal, a part of the epidermal tissue. Now the stomata is of 4 types. We have the ranunculaceous or anomocytic type, the cruciferous or anosocytic type. We have the caryophyllaceous or diacitic type. We have the rubiaceous or parasitic type. We have the graminaceous type, which is only found in monocots. Stomata are used for the function of exchanging gases between plant and atmosphere. Evaporation also takes place through the stomata. Hairs or trichomes. These are also part of the epidermal tissues. Some epidermal cells of most plants grow out in the form of hairs or trichomes. They may be found singly or less frequently in groups. They may be unicellular or multicellular and occur in various forms. The cells of the hairs may be dead or living. The hairs may be several types, as stinging hairs, lacciferous hairs, and so on and so forth. The trichomes may be classified into different morphological categories. One common type is referred to as hair, which may be divided into unicellular or multicellular. Trichomes control the rate of transpiration. They also reduce the heating effect of sunlight. They aid in the protection of plant body from outer injurious agencies. These are what they look like. Different trichomes. And we have the hairs on the epidermis on this plant shown here. The other tissue system is the ground tissue system, which is found between epidermis and vascular cylinder. It's called cortex. In vascular systems, the central tissue may be called medulla or pit. In monocots, there is no delimitation for ground tissue. That means the cortex or pit are not different. They are mixed together. Cortex, this is the ground tissue that is found between the epidermis, which surrounds the central cylinder, and is delimited from the cylinder by the endodermis. Consists of thin-walled parenchymal cells having sufficiently developed intercellular spaces among them. Some of the cells maintain their chloroplast at least at the young stage. It also contains starch and so on and so forth, but it may contain cholenchyma, sclerenchyma, and sclerites in addition to ordinary parenchyma. The cortex acts as a protective tissue, but secondarily, carbon assimilation, storage of water, storage of food, and also other functions are also carried out. Cholenchyma of the cortical region aids in the temporary mechanical support of plant body. In roots, it is a storage tissue and helps in pumping water from hairs to xylem. The pit or medulla forms the central region of the stem and the root. Usually, the pith of dicot is largely parenchymatous. It is devoid of chlorophyll in the mature state, but starch-forming leucoplasts are found in it. In monocot stems, the vascular bundles are scattered throughout the ground tissue, and the pith is not distinguishable. The extensions of the pith in the form of narrow parenchymatous strips are called medullary or pith rays. Functions. Pith cells mainly serve as storage tissue. In case the pith is sclerenchymatous, it acts as mechanical tissue and provides mechanical strength to the plant. The medullary ray consisting of parenchyma cells serve as channels for the transport of food materials and water from the central part to peripheral region of the stem. So in dicot, this is the pith is defined. This is the cortex. Okay, immediately after the endodermis, epidermis, in between the vascular system and the epidermis, you have the cortex. The ground tissues. So we have the cortex, which is made up of collenchyma and parenchyma, while the pith is in the middle for the dicot stem. However, for the monocot stem, the ground tissue is not differentiated, okay, because the vascular system is scattered. Okay, the vascular bundles are all scattered. So the ground tissue for monocot is not differentiated, unlike that of the dicot, which has its delimitation where it is existing, from the vascular bundle. But this one, you have them scattered all over the transverse section of the stem. The vascular tissue system. The third tissue system is the vascular tissue system, which consists of a number of vascular bundles which are found to be distributed in the stele. The stele is the central cylindrical portion of the stem, and the root, commonly surrounded by the endodermis, and consists of vascular bundles, pericycle, pith, and medullary rays. That's make up the stele. Okay. Each vascular bundle consists of xylem and phloem tissues with or without cambium. The function of this system is to conduct water and other nutrients from roots to leaves through the xylem and translocation of prepared carbohydrates from leaves to other storage organs and growing regions of plant body through the phloem. The vascular bundles may be arranged in circular ring as in dicotyledonous stems, and the roots, on the other hand, they're found to be scattered throughout the axis in the monocot stems. Constituents of vascular bundle, are made up of xylem, phloem, and cambium. Now let's look at the xylem. The xylem lies towards the center and is composed of vessels, tracheids, wood fibers, and xylem or wood parenchyma. The xylem elements aids the uptake of water and mineral salts from the roots to the leaves, while wood parenchyma are living tissues and aid in storage. The wood fibers give mechanical support to the plant body. Phloem or bast. Phloem is found away from the center of the axis towards the periphery and consists of sieve tubes or sieve elements only, or sieve tubes and companion cells only, or sieve tubes, companion cells, and phloem parenchyma. In gymnosperms, it is represented by sieve cells only. In most monocot, it consists of sieve tubes and companion cells only, whereas in dicot, sieve tubes, companion cells, and phloem parenchyma possesses simple pits in their walls, particularly which lie against the sieve tubes. So phloem serves for translocation of prepared carbohydrates from leaves to the storage tissues and other growing regions. Sieve tubes translocate proteins and some other carbohydrates. Phloem parenchyma conducts amines, amino acids, and soluble carbohydrates, and companion cells also translocate many soluble food materials. All phloem elements are living and formed of cellulose. In certain cases, the primary phloem is capped by a patch of sclerenchyma called hard bast. Cambium. In between xylem and phloem, a thin strip of primary meristem is found in dicot stems, called cambium. The cells of cambium are rectangular and thin-walled. The cambium strip may be uniserrate or multiserrated. The cambium cells are living, sufficiently elongated, and possess oblique ends. but as they become flattened tangentially, they look rectangular in cross section. Okay, these are the xylem and phloem elements or cells. We have the labels, we have the vessel member, we have the perforation plates where the water flows through from. You have the tracheids, fiber, which gives it that strength, the phloem as well. Fiber gives it that strength. We have sieve areas, we have sieve plates, we have companion cells. So that's all for this lecture. We will be looking at let me just draw back to this. You see the vascular cambium in between the xylem and the phloem. Okay, so that's what we're talking about. The xylem, cambium and phloem make up the vascular tissue. So that's all for this lecture. Feel free to subscribe and drop your comments in the comment section. I will respond to them immediately. Thank you.